So working with the Miller Lab at Rice University, we were able to fabricate these structures using living cells and then perfuse them with blood to test their fluidic properties and ability to oxygenate. So we're just seeing this thing being pumped with air and then perfusing it with blood cells.
There were some like cool unexpected behaviors where you get a kind of mixing within some of these blood vessels, and there's kind of some speculation of whether or not that is like biologically accurate or biologically useful in terms of increasing oxygenation. And this is sort of the scale of the 3D printed alveoli analog that we created. And we're still sort of, early work in this future of creating actual organs that could be transplanted into humans. And this structure is still more than an order of magnitude larger than a real, lung alveoli.